Right. Thanks everybody for joining. We've got Heloise here. She's somebody in the community and has been really involved with volunteering throughout, I think the last decade or so. And she's doing research as well. Part of her team at Stanford has been helping develop a cure for FSHD. So just here to kind of get her insights on the research space and what she's working on specifically. So Heloise, if you want to expand on that bio a little bit.
Yeah, hi everyone. Let me see if I can share my screen. Cool. Can you all see that?
Yep.
Yep.
All right. So I'll just talk with you guys a little bit today about my journey with FSHD and how I came to be involved in research, and also touching a little bit about all the hope that's been going on in the field. And then after that, we can just chat a little bit. But um, without further ado, my name's Eloise. I'm currently a junior in university studying bioengineering, research, and medicine. And ever since being diagnosed with FSHD, I've really made it my mission to continue advocating and researching for treatment until there's an affordable and effective treatment through the veins of every single patient around the world. And so it wasn't, a very linear path, as I'm sure is the case for all of you guys. I was diagnosed at 13 years old after about 3 years of searching for a diagnosis, feeling like a guinea pig, running every possible test, and being being misled by physicians that I trusted, until finally I got the diagnosis. I was the first in my family to be affected with FSHD, spontaneous mutation. So no one else in my family has it. And so these four letters, FSHD, were really new to me. And it was extremely isolating to be diagnosed with this disease, as I'm sure, we've all gone through. And the next 2 years after my diagnosis, I went through a really, really dark period. And I didn't think that I would ever recover until I met June, who I can really say changed my life when she introduced me to the FSHD community and told me about the research and hope that's been going on in the space. And I guess little by little after that point, I started to open myself up a little bit. And until I finally decided to set my foot down and take action, and became really involved in the community. And so ever since then, ever since the first, so I ended up founding the Southwest Florida chapter because my family was living in Florida at the time. And ever since that first chapter meeting, something in me, just a light switch, went off. And I like to say I became addicted to activism, and I feel like there's just so much strength in our community, and FSHers are some of the most driven and adaptable and resilient people that I know. And so it's been really transformative and empowering to be involved with the community. And so that's just a little bit about my background with advocacy. And ever since coming to university, I've been exposed to a lot of like bioengineering and biochemistry tools, which have been really exciting to learn about and to think about how to apply them to FSHD. And so without further ado, I'll give a short talk today about first kind of recapping the science behind FSHD, which I'm sure you've heard 1,000 times, but it doesn't hurt to go over it once more. And then highlighting some of the current therapeutic approaches that are in clinic. And then finally, I'll talk a little bit about the research that I've been working on. But so first things first, what is FSHD? I mean, we all already know what it is, but I guess from a clinical standpoint, it's a form of muscular dystrophy causing progressive weakening of skeletal muscle. And something that's really interesting about FSHD and really uncharacteristic and uncommon, I would say, is how differently it affects each patient. Um, Not only the asymmetry within one patient's own body in terms of which muscles are affected, but the progression is very nonlinear. It's really difficult to predict. And so that kind of comes with its own challenges when it comes to clinical trial design and drug studies. And it means there's a lot still to be learned and discovered about the molecular mechanism behind FSHD. But the good news is that we know enough at this point in order to be able to try to treat FSHD. And so there's a lot of hope in the scientific space. And then I also wanted to mention early onset FSHD, which I'm sure a lot of people in this group are familiar with and maybe intimately familiar with as well, in which symptoms typically come in the first 10 years of life and are accompanied by really rapid and severe progression. And so I'm really glad to see that early onset FSHD has been getting a lot more attention recently, but there's a lot more work to be done in order to include pediatric populations in clinical trials. And so moving into the kind of molecular mechanism behind FSHD, I'm going to start by recapping this kind of central dogma, which is often so the DNA is the kind of library of, and it contains all of the information necessary to build the building blocks of life. And that's the really amazing thing about DNA, is that it contains all of the information that you could possibly need for life. But that doesn't mean that all of those genes or all of those little bits of DNA should be on all the time. But then DNA gets converted into RNA through this process called transcription. And RNA is a very similar molecule to DNA, but it basically acts like a messenger and a transcript of the DNA. And then the RNA gets produced into the final building block called a protein, which is what performs all the functions of life through a process called translation. And in order to kind of boil that down into an analogy that I'm sure you've heard before, you can think of the DNA like a huge library with you can think of it like the library of the Culinary Institute that has all of the cookbooks that you could ever need to make any recipe that you could ever want. And there are some sections of the library that are, open access, and you can take those recipes whenever you want. There are some sections of the library that should be always, off and restricted access only, and some portions of the library that should be open sometimes and closed other times. So there's a lot of regulation that happens in this library. And then you can think of the RNA molecule like a transcript of that of a specific recipe that you want. So let's say you wanted to make gingerbread cookies. You could go into the DNA library and jot down the recipe from the cookbook to bring home with you, because you don't want to have to bring home the cookbook every time in case it gets lost or damaged. And so that's why we make a transcript of it. And then you can think of the protein like the final finished product, in this case, making gingerbread cookies. But what happens when, instead of making these festive gingerbread cookies, we make an evil, toxic protein instead? Well, that's what happens in the case of FSHD with a protein called DUX4. And so, as I'm sure you've heard before, FSHD is caused by expression of the toxic DUX4 protein, which is actually, it's important to note that this protein is actually supposed to be used in the very early stages of development, during embryonic development, but it actually is supposed to be permanently shut off or silenced in adults after the embryonic stage. And because of a genetic mutation in the DNA, DUX4 stays on. And so that's kind of like the root cause of FSHD. Now, DUX4 is part of a family of proteins called transcription factors, which essentially are just responsible for regulating when certain other genes gets expressed, so when other recipes get produced. And so DUX4 is supposed to be off in healthy people, but because of a genetic mutation in FSHD in the DNA library, DUX4 gets continuously expressed, and it has hundreds, if not thousands, of other gene targets. So basically, when DUX4 becomes expressed in the skeletal muscle of people with FSHD, it will go and turn on all of those targets that are supposed to be off. And so you can imagine if you have this kind of like peaceful library where people are coming in and out as they need, but then all of a sudden you have this toxic DUX4 protein that comes and like turns on all of the genes and like opens all the cookbooks and sends them all to the kitchen to be produced. That wreaks havoc in muscle fibers, and that's what leads to the degradation of muscle seen in FSHD. And if you're curious to see, on the right here, you can see um, a cross-section of healthy muscle fibers versus cross-sections of muscle fibers with FSHD. And so this expression of the DUX4 protein is what is the root cause of FSHD that causes muscle degradation. Now that we've covered kind of the molecular biology behind FSHD, we want to understand what will it take to get a cure to the clinic. And this is a really nice figure put together by Peter Jones's group, if you're familiar with his work. And it kind of covers all of the different approaches that you could take in order to stop the progression of FSHD. But there's a lot going on in this figure. And so I kind of broke it down into different pieces. So if we go back to the analogy with the DNA library, we have a shelf here with the DUX4 gene, which is supposed to be locked up and silenced in people with FSHD. And so one potential approach to stop DUX4 from being expressed is to just put a lock back on that gene because of that mutation, the lock has come off in people with FSHD. And so I'm going to talk today about the companies that are currently in clinical trials, but I want to preface by saying that there's a lot more work being done kind of behind the scenes, and a lot of other companies are working on different approaches that might be still in the preclinical stages, or that they haven't gone public yet. And so the approaches I'm talking about today are just a sliver of all the excitement that's happening in the field. But so this kind of idea of putting the locks back onto the DUX4 gene is what the company called EpiCRISPR is doing. They used to be called Epic Bio, and they've actually just I think just gotten clearance from the FDA, if I'm not mistaken, to start clinical trials for FSHD using their approach. Then the next step, we could potentially make an intervention at the RNA stage, or that kind of copy of the recipe. And so we could put that copy through a shredder so that the DUX4 protein never gets produced, and so that the recipe never gets sent to the kitchen and the DUX4 protein never gets the chance to be produced. And this is the approach that's being taken by Arrowhead and Avidity. And this is a really exciting approach as well that's currently in the clinic. The last stage of the evil DUX4 protein cookie that's expressed, you could potentially think of ways to cut up that protein so that it doesn't have the chance to go wreak havoc through muscle, like we talked about. I don't know of any companies that are working directly on this part of FSHD, and correct me if I'm wrong, June. But I don't know if there are any companies currently in trials for this. But there are also interventions that could be made to repress the pathway, the gene expression pathway that comes after DUX4 ultimately is expressed. Like, even if the toxic protein DUX4 is expressed, what can we do to stop it from having negative impacts that degrade muscle? And Roche is working on an adjacent field to this, and I think, again, correct me if I'm wrong, June, but I think they actually have like 2 different trials going on.
Yeah, no, that's correct.
Yeah, so
do
their
you want me to
approaches are
talk about
rooted
them?
both on the regeneration of muscle and also on repressing like a part of the DUX4 pathway.
They have a therapy that aims at the immune response
Mhm.
occurs that can maybe worsen the development of the disease.
Yeah. And so that's another way that companies could approach FSHD And that's also what my group has been working on with Stanford iGEM. And so perfect transition into introducing my team that I've been working on over the past year. So I've been working with a program called iGEM, which essentially gives the framework for undergraduates to perform research on synthetic biology. And so over the past year, I've been working with this group on an FSHD research project for a potential novel therapeutic approach. And it's been a really rewarding experience, and I'll dive into that in a second. But now the project is kind of going on to the next phase, but this is kind of the group that the project started with. So to give you an overview of the project, if we dive in one level deeper into what does the DUX4 protein actually look like, this is a very simplified representation of what the protein looks like, but it basically has these two primary parts to it. It has this DNA binding domain, which identifies and binds to specific target genes in the DNA. So it goes and looks for specific cookbooks or recipes in the DNA library to attach to. And then the second part, the TAD or transactivation domain, is responsible for activating those genes. So in the context of the analogy, this part of the protein would be responsible for actually writing down the recipe and making the copy of the recipe. And so this is kind of the basic structure of the DUX4 protein. And this full-length DUX4, when it has both these DNA binding domains and the transactivation domain that actually turns on those genes, this whole protein is what we call DUX4 full-length, and that's the protein that's toxic to skeletal muscle, and the protein that's the root cause of FSHD. So building off of that, what if we could stop DUX4 full-length, or that toxic complete protein, from both binding to its target DNA sites and also stop it from turning those target sites on? And so that's kind of the crux of what my project has been working on. And I've been really excited about the kind of preliminary data that we've been gathering. It's essentially centered around around engineering these new proteins that feature the same DNA binding domain as DUX4, so that it'll bind to all of the same sites. And going back to the analogy, go open all of those cookbooks, but instead of actually turning on those genes, or instead of writing down those recipes, it'll actually like lock them up. And so that's kind of the project that I've been working on over the past year, and I'm excited to see where it goes. And I want to thank all the people that have been working on it with me, notably the Stanford iGEM 2024 team, who you saw in that photo a few slides back. And I'm currently working with Dr. Stanley Chi here at Stanford on the epigenetic mechanisms. And I'd also like to thank all of these other mentors that have contributed to the project. And so that was kind of an overview of my journey, the science behind FSHD, and the research I've been working on. And so thank you so much for being here. I'll stop sharing. Yeah, and I'm happy to answer any questions about the project or the journey, or also happy to just chat for the next bit of our time together.
Thanks. That was a really helpful overview. One question. So what led you to, like obviously there's a lot of different ways to target the DUX4 and downstream effects. What led you to kind of focus on the area that you chose to?
Yeah, really good question. So there, as you mentioned, there are a lot of there's a lot of excitement in the field, and it's crazy to think that, just over 30 years ago, we still didn't know what gene was causing FSHD. And now we know not only what that gene is, but how to stop it. And we know that there are a lot of different ways to go about stopping it. And so I guess like what's interesting or unique about this approach specifically is that actually, as June mentioned, there's a lot of inflammatory response and immune response involved in FSHD due to the pathway that DUX4 activates. Essentially, when DUX4 is expressed, a lot of inflammation and immune effects happen in and around muscle fibers. And so essentially, when you deliver from a clinical standpoint, when you deliver kind of external parts that the body doesn't recognize, external genetic material or external proteins, that could actually potentially contribute to some immune effects in the muscle as well. Although that's not, proven. There's some data that suggests that. And so I guess we're kind of like diving into more of the molecular biology now, but this approach is rooted in the expression of and the delivery into muscle of proteins that are fully human and fully derived from existing human protein domains. And so basically, like your immune system, when it sees those new proteins, won't freak out and send an inflammatory response to the muscle where the therapy has been delivered, is the idea. Of course, like we're still in early stages, so we haven't shown that to be the case yet, but that is kind of like one of the primary um, advantages to this approach, and then, or one of the primary motivations for this approach. And then the other aspect is thinking about a delivery standpoint. The genetic sequence that encodes for these engineered proteins is fairly short, and there's a certain type of packaging method called an AAV, which you can use to basically target skeletal muscle. But it can only take like a load of a certain size, and so the advantage is that the size of this protein would actually like fit within this delivery vehicle, and that's kind of the extent that I'll get into it. But
Well, since you raised AAV, have you thought a lot about the different delivery vehicles for this type of therapy? Because there are challenges with using AAV. Well known is that it can, well, it tends to accumulate in other tissues besides muscle to the extent that it can be quite toxic. If you're trying to get enough in the muscle, it can accumulate in the liver. And as you know, Kate Therapeutics has developed an AAV that is more muscle specific. So that's been interesting and exciting for people working on muscle disease. But as a virus, it also induces its own immune response from the body. So it's generally thought that if you take an AAV-based therapy, that you just have that one shot. You take it, but then after that, your immune system will reject any additional therapy using that AAV. So it's potentially a little risky that way. And then, of course, a lot of people already have immunity to AAV because it's a common virus. So if they already have that immunity, they might not be eligible to take treatment using an AAV vehicle. I'm just sure I'm telling you things you know, but maybe the rest of the audience maybe is less familiar with these issues. So I was just, and people are working on other types of like lentivirus, or even I think lipid, like non-viral molecules, little ways of packaging the therapeutic molecule and getting it into cells. So I'm just curious if you and your team have, had conversations about these issues as well. What your thoughts are.
Yeah, those are all really good points. To address kind of the first point about the tropism of the AAV, or like where in your body it will gravitate towards. In general, AAVs have, and I'm just to preface, I'm not by any means claiming to be an expert in AAV or anything like that. And other approaches for FSHD do use AAV, and so and for clinical trial purposes and to make it to the market, like any of these therapies that do use AAV will undergo rigorous testing to make sure that it's safe and effective. So just wanted to preface that their AAV technology is fairly recent, but in order to be approved for treatment, it will have to go through rigorous safety and efficacy training. So as you mentioned, Kate Therapeutics has developed this kind of new strain of AAV called MyoAAV, which selectively targets skeletal muscle, which is kind of like primary the primary delivery method that my team has been thinking about. That being said, we have also thought about a lot of the challenges that are still associated with it, and so that's definitely a conversation that's been going on. But right now we're still in the optimization stages of the construct itself. So I guess like with any therapy, there are two parts of it, right? The actual drug that will have an effect in the skeletal muscle, and the other part is the delivery vehicle. So how do you actually get that drug to the skeletal muscle? And so right now we're still kind of in the first phase of optimizing the construct or the drug that will have an effect in the body, or like in the muscle to stop the DOX4 activation pathway. But the delivery question is something that's kind of in the back of our heads as well.
And I might have missed this. Are you moving into in vivo, like animal models soon?
Yeah,
or-
so that's kind of the hopeful next phase of the project. We're not there quite yet because we are still working on optimization of the construct, but we are gearing up for in vivo trials, which is exciting.
Cool. Do you have in mind which mouse model you would use?
Yeah, I'm not sure how like technical we want to get in this meeting, but we've been thinking about that.
Okay. I don't want to monopolize the questions. If other people have questions, please ask away or use the little hand raise thing in Zoom.
I think I saw previously that you were looking at stem cells as well, like as a combination therapy. Is that something that you saw progress with?
Yeah, that was that was like along in the early stages of ideation. The kind of like ultimate dream is to address kind of a combination therapy, that because with FSHD, there's one side of the coin that's stopping progression of the disease, and the other side of the coin is regeneration. And those have often been looked at as two separate issues, which is a necessary starting point, because a lot of these new biotechnology and bioengineering tools are really recent, and the technology isn't quite there yet to make something that addresses both. But that being said, I think bioengineering is moving in that direction right now as a whole field. And so the ultimate goal would be to be able to develop a combination therapy. So yeah, I think, I'm still an undergraduate, and so there's still a lot of time ahead, but that's something that I've been thinking about as a potential like grad project.
And then a few others like Avidity, Arrowhead. It sounds like their approaches are all pretty similar with different types of RNA interference. If we saw kind of disappointing data for one of them, would that, do you think, give a negative outlet for the others? Like, would that be a pretty close relationship between them?
Do you mean like if one of those were to
Yeah, like
succeed?
like a readout from one of those three, because they're pretty similar, was negative. Do you think that that would, like in your view, prematurely?
Oh, like if the readout was negative from one of
Mm.
them, would it impact the development of the others?
Not necessarily impact the development, but like, would you then kind of not have a strong expectation for the results of the others? Like they may still progress the trial, but you just wouldn't expect it to be too effective.
I think it's hard for me to comment on that because I don't know, like what their data looks like, and I don't have like strong insights into what the biotechnology world looks like on the kind of commercial side because I'm very much more on the kind of like academic basic translational science side of things. But all I can say at this point is from the limited data that, for example, Avidity has already shared publicly, um, it looks like they're having um, positive readouts, at least in these early preliminary stages. That being said, drug development is a really arduous and competitive process. Man, I'm taking a class on this and we just it's like 95% of stages of drugs that make it to phase 1 trials don't end up like making it all the way through, um, or something like that. Or yeah, I might be butchering that number, but
Like, yeah, maybe 1 in 10 or 1 in 20 actually get to approval, or
yeah, exactly. And so I mean, I don't want to, I can't predict the future of what the success will be like for those companies, but I do feel extremely hopeful. And I mean, yeah, we in the field of bioengineering, like we are moving at unprecedented speed. And there's just so much interest that has ramped up for FSHD over the past couple of years. Um, in part because it is a really interesting disease scientifically, and I think largely in part due to the strength of our community and advocacy efforts. And so I think there's every reason to be hopeful that there will be an effective treatment over the next years. As to who exactly is going to reach the finish line first, who knows? Because of course, we all felt a huge blow when the news from Fulcrum came out, that they were discontinuing their phase 3. And so, I think we have to be prepared for news like that, but I think we also should be primarily prepared for some hopeful news over the next years as well.
I don't know enough about the differences between the vehicles that Avidity and Dyne and Arrowhead are using. And also, Miracule also is using basically an mRNA-based, strategy. But so much, there's so many factors involved in making it successful, even just from a purely mechanistic, leave aside all the business complexity of, business in developing drugs, but just how effectively does it find muscle, get into the muscle, how stable is the molecule inside the cell, how effectively does it bind the messenger RNA, and so on. I think all these can be really affected by very subtle, differences in the molecules and the antibodies they use. And I imagine because of the, each one of them is patented, they do have differences. So in order to be a unique product that they can protect. So at the end of the day, it really comes down to how does it perform in the clinic and in terms of the adverse events that might happen, and the efficacy of the drug. And if a drug goes through clinical trials and it doesn't meet its endpoints, it's not you can't simply conclude that the drug didn't work, that it didn't engage its target. And there could be so many other factors. It could be that several of the sites where the trial was conducted were not as rigorous in how, or in the way they did the assessments, or that one of their piece of equipment wasn't calibrated correctly, or I mean, they're supposed to control for these things, but they do happen, and so or that the outcome measures they used just weren't sensitive enough. They weren't quite the right ones. So people are debating that right now with what happened in the Fulcrum trial, for example. We don't, you know, I mean, that's a very mysterious situation, because we saw some positive clinical outcomes in phase 2, and we continued to see them in the open label extension. So these are people who are on the phase 2 trial that once the trial ended, everybody had the option to now go on the drug, and they've been on it for, I forget, 96 weeks, or over more than 2 years, and past the clinical trial. And that study showed that, first of all, the people who were on losmapimod, or who were not, had been declining when they went on the drug. They started to improve. So they started to look like the people who were on the drug. And over more than 2 years, they all continued to do pretty well. They were stable or improving. So how do you explain that in contrast to what they say they saw in the phase 3? And we haven't really seen all the phase 3 data yet, too. So, how was the phase 3 different from the phase 2? It's all, one thing that Jeff Statland recently presented at the Muscular Dystrophy Association, and actually next month he'll be giving a talk for the FSHD Society. He'll do a webinar and present the Fulcrum phase 3 data. So I hope you all come to that, because it's quite interesting. So in the entire, cut me off if I'm going to, I geek out on this stuff. I'm so, because I I live it and think about it, and what does it mean? So but in the phase 2 trial for Fulcrum, to qualify, you had to have an MRI that showed that you had a muscle that had excess water in it, which we interpret to mean that there's inflammation, because that's the muscle gets edema, it gets fluid. And so you had to have that, and you had to have like a needle biopsy from that muscle to be in the trial. So what this meant is they selected people who all had active disease. They had a muscle that was actively degenerating. And phase 3 trial did not have the MRI qualification. So it was presumably a much more diverse population in terms of whether they were actively in a disease state, or as we all know in FSHD, it can kind of go quiet and be sort of plateau for a long period of time. So you'd have to think that that could have a major impact on the data. When you average everything from phase 3 trial, you're looking at what probably is a very different population than who you looked at in the phase 2 trial. So the way the phase 3 trial is done, you might actually miss a drug that actually does help when somebody is in an active disease progression state, so these are the things that, like, really, we're trying to figure out, how do we figure that out? Could we possibly do another trial? Someone else would take up, adopt losmapimod and do a more rigorous, or a different kind of trial based on what we know so far. So that's just an example of how complicated this is. These are science experiments that with a lot of different factors you have to consider. And anyway, I just wanted to
I mean, to add on to that, I think, I mean, the space has been talking a lot about how we learned a lot from the Fulcrum trial in terms of like what readouts don't work, what readouts work, inclusion and exclusion criteria, and it sounds like they've been really generous with their data sharing too, and their procedures. So I think that goes to highlight too the kind of collaborative nature of the FSHD space, and how, even though we're all devastated that this trial didn't work out, it will hopefully be able to inform stronger and more rigorous trials in the future. So
And yeah, what Fulcrum has done is in agreeing to share their data is really unusual. You would hope that it would be the norm for companies because they really don't have any use for this data, but after trial's over, but it's unusual, and I think it speaks to the strength and outspokenness of the patient community. I mean, that people have really put a lot into developing relationships and having their voice heard. And that puts a lot of, well, it's not only pressure, but I mean, I think, it makes companies like Fulcrum feel, that they need to do whatever they can to continue to benefit the community. Uh, so, so I guess it's a way of saying like, y'all, like, be active, be engaged, because it does matter. It makes a difference.
yeah, I think one thing that's been really sparking me recently is the fact that as individuals we actually have so much power in the whole process. I feel like drug development can often feel like this big, like, black box um, from the patient perspective, and like we don't really know what's going on behind those closed walls. Um, but I truly believe that by using our voices um, and like pestering everyone, like in whether it's calling senators to get them to approve funding for FSHD, or whether it's making sure that clinical trial that biotechnology companies know we are out there and we want a treatment as early as possible. Whether it's raising awareness in the community so that more even so that more doctors are aware that FSHD is a thing, and to help people who haven't received their diagnosis yet. Um, yeah, I think we have so much power as individuals just by using our voice, which doesn't take much except for some hope. So
For your research process thus far, what would you say has been the most difficult part?
mm. I think it's definitely been a huge learning experience along the process too. I mean, I had some lab experience and like some science project experience from before, but this was definitely a huge leap in terms of like independence and project design. So that was definitely a large challenge at first. I guess from like a project management standpoint. Um, I think one really exciting part is I remember still very vividly like the first day that we got some real data after like a couple of months of like getting the cell culture conditions just right, and like cloning, or which is the name for the process of like making like certain DNA constructs. And like after a couple of months of prep work, we finally were able to put our DNA into cells, and then take the cells out and look at them under like a plate reader, which basically quantifies fluorescent expression, which is how we were quantifying um, the efficacy of our construct, and like seeing those real results come up on the screen was just so empowering. It was like, wow, like there's a potential cure in my hands. It's still very far off, and I think there's a lot of exciting stuff happening in the meantime. But it is a really cool way to take back control, and I think that's what kind of like pushes me through the days where maybe like my cells aren't growing well, or it looks like the data's a little like like I'm having to tease apart the data a little bit. Yeah, I feel like just like the hope and the empowerment that comes from this project is really what like keeps me pushing through. Because basically like the cells, I'm currently in still in the in vitro phase of the project, um, working on patient-derived cells, which are derived from which basically like wow, bioengineering is so cool. But I'm using induced pluripotent stem cells, which are essentially like, say a patient got a blood draw, a patient like decided to donate tissue, to donate blood to research. Those blood cells were reverted back into stem cells, which are like the earliest stage, the earliest cell type. Um, and and then what I'm doing now is taking those stem cells and differentiating them so that they become muscle, and then um, cells will like endogenously express DUX4 because they are from patients with FSHD, and then would be able to test all of the um, constructs on those cells. But working with these stem cells is a lot of work because you have to take care of them like every day, and if you like let them growing even just a couple hours too long, like they'll start to randomly differentiate into random cell types, and so they're very finicky, but it's okay because yeah, I feel very very driven.
I remember that made an impression on me when I had a summer when I spent in a biological lab. So I studied physics. So I always think of experiments as like you go in when you want to, you turn things on, you turn them off, you walk away, you come, and then the lab with the living cells, you go like, uh-oh, once you start something, like it's on its own schedule. And if I have to stay up all night to do things or, you know, yeah, you have to, it's very different.
The cells don't care that it's, you know,
You
11
have a
p.m. on a
final exam
Sunday.
and, you know, like, whatever. So you have to
But there
plan.
are there any key ways that this community or the broader like FSH community that watches this can help you, like, support your efforts?
Oh, um, I would say use your voice, especially in these times. We need funding for FSHD more than ever. We have so much momentum, and we're almost at a tipping point for FSHD, and we can't, you know, scientists and labs can't continue the important work that they're doing if they don't have funding to continue those projects. And so as an individual, as individuals, we have so much power to use our voice to advocate not only to the government, but to the communities close to us about why funding is necessary for science. Um, it's something I've been thinking a lot about for this project. And I think also just like from a personal perspective, using raising awareness about your story is really empowering, and helps you take back control of your story. And so I think yeah, just use your voice. As Ali Rhodes says, get angry, um, don't back down. I think that's truly the best way to keep pushing the needle forward.
So as you move forward, I mean, where you wonderfully won, your group won quite a nice chunk of cash prize at the iGEM event in Paris to, I guess, you use it to fund the research, I suppose. I guess if you wanted, you could have used it to take a vacation or something, but, and
Oh, just to clarify, this it's not I could not use it to fund a vacation.
had to use it for research.
After like outside of the scope of iGEM, we participated in a program through Stanford, through which we won like a seed grant for the project. So
Yeah. Okay. So, and so is that going to fund the work through the coming year, or do you have to raise additional money? Are you kind of thinking about
yeah,
where?
so we won we won that grant in December, and at first through my naive eyes, I thought that it would, last us over the following year or two, until I realized that science is really expensive, and even like the little pipette tips and like the media or like the food that you use to feed your cells, which I have to go in and change every single day. One bottle of it that's like this size costs like $600. And so science is really expensive. So I've been starting to look into like I think the money that we have will last through the rest of our in vitro experiments, but moving into in vivo, we'll have to look into alternate funding. And so I've been starting to look into other sources of grants for that.
Yeah. I did tell you about IndieBio, right?
I don't think so.
Um, I think they're kind of a biotech incubator in the Bay Area.
Hmm.
And you apply to be taken into their program, and it comes with startup funds, and then it ends with an opportunity and coaching along the way, and then you pitch your idea to a bunch of Silicon Valley investors at the end of it.
I would appreciate if you could send me the info for that. Yeah.
Okay, I will.
We've been working through an institute at Stanford as well called the Innovative Medicines Accelerator, which is kind of like a similar idea.
Yeah, well, Stanford ought to, yeah, they should have plenty of resources and connections to help with that too. Um, but yeah, I'll just
And just want to make sure if anyone else has questions, feel free to ask or post in the chat.
I'll send you an email with IndieBio. Right.
Oh, Zach, just seeing your message. Thanks for your question, KP. Yeah, I'm really interested also about that same question. So I'll check out that video recording.
And then Del, I know you're a physical therapist. I think you're on that call as well. If you want to make any quick comments on that first question in the chat, feel free. If you're able.
Let me check.
Oh, hi, Andel. Are you out of the car now? I can see you.
Oh, yeah. Yes. Actually, the phone was in my pocket the whole time for like
Oh,
the last like 10 minutes eating dinner.
oh, okay. Well, don't be shy. We're all used to seeing each other lunch, breakfast over
What
Zoom.
role played by exercise and what percentage by diet? I mean, I can't give true specific numbers, but I can say for myself, I know when I have, let's see, foods that have a decent amount of sugar, it definitely agitates me. Um, Basically, if I try to eat more stuff that's on like an anti-inflammatory type of diet, I tend to feel better. So that works for me significantly. I can't talk about it from a truly scientific standpoint, but I definitely feel as though eating less sugar and from a anti-inflammatory vegan type of diet, I definitely do better. Exercise, it's kind of like all the other muscular dystrophies. You have to, toe a fine line between, because every patient is different, at different levels. And so you have to do enough to, help them exercise, but not to the point where they regress. Right. And so that's something you and your physical therapist or occupational therapist have to figure out together, or even with a personal trainer, you have to figure out together, what's that fine line. If you want to say, okay, bicep curls, I can do 2 pounds and I'm good 3 sets of 10. But I know if I do 4, my arm doesn't work properly for like 2 weeks. So okay. I know that's a very like simple example, but you just have to kind of feel it out, trial and error as to how your body is doing, especially in that moment too. Because as we do know, with FSHD, you have periods where you just had a plateau, and then next minute you're kind of regressing a little bit more, so then you have to kind of reassess. So, and obviously, if you exercise too much, then of course you have a large inflammatory response, and then the muscles get inhibited, and you start to say "ow" more. So, exercise is good. You just have to figure out what's good for you. I've seen some people, they can do high intensity exercise, lift weights, they can run. I cannot. That doesn't work for me. If I do that type of stuff, I'm inflamed and then I can't go to work. So everyone's just a little bit different. So that's something you have to figure out with your physical and occupational therapist. What exercises work right for you at what frequency and stuff like that. But I definitely think eating healthy plays a big role. Less sugar, less meat. Definitely, like an anti-inflammatory type of diet goes a long way in helping. Hope that answers that question.
Yeah, that was really good. On the point about the inflammatory response post kind of strenuous exercise, do you think a general rule of thumb would be if it's a more affected muscle, probably go mild with like go light with it, and then if it's less affected or not affected, then you're safe to use it more.
Yeah,
Is that
for
what
the
you think?
most part. For the most part, yeah. I mean, based on what you tell your therapist and what you've done in the past and what is too much for you, they can create a program and try to have an idea. Okay, all right, I think this might be too much for you, but let's try this. But it's only until you actually try where you'll actually see, okay, what works and what doesn't work.
Yeah, I think there's definitely a lot of trial and error and paying close attention to how you're responding to it. And I think working with a good trainer or physical therapist to assess which of your muscles, because they're not all affected in the same way within a particular system that you need. Specific muscles in there could be degenerating and other ones around it might still be healthy. And so,
Yeah, very
so,
true.
and it's hard, I think, for individuals to do that assessment themselves unless they become very expert in their own own muscles. But I don't know, Zach and Eloise, what has been your experience in terms of kind of figuring out what exercise works for you?
For the most part, I just kind of do it and don't really have too much of an issue after. So, like some things I obviously can't do, like with the shoulder, but recovery times for the most part are fine.
Mm.
Yeah, I think it definitely varies day by day. And I kind of have to feel out what my body needs and doesn't need. It's a very hard line to toe because if I, don't move or don't exercise, I feel like I start to get a little jittery. Um,
Mm-hmm.
and it affects like my mental performance, but then sometimes I'm just like too weak to really move around, and so that's kind of a frustrating gray area. Um,
Yeah.
but then, yeah, I mean, there's like some things that have sailed a long time ago, of course, but then it really comes down to day to day. But I think I'm starting to like figure out a routine. I personally, if anyone hears this in the future or is interested now, I personally really like Pilates because
Mm-hmm.
it is a very gentle way of working kind of like all of the muscles in your body and some muscle groups that you don't typically use in your day-to-day routine. And I've found a trainer that um, I was able to give like one of the FSHD physical therapy flyers to, and so she's been able to work with me to really kind of like adapt um, to exercises that I can't do and leave out the ones that are too intense. So that's something that
Nice.
I've found works for me, but it might not work for everybody.
Mhm.
Right.
Last
I
time, just real
definitely,
quick, any final questions?
Definitely move, because when you don't move, then the body tends to not want to, stay lubricated, and it just doesn't function right. So at least move a little bit each day.
that sounds like good advice. I've heard other people say that cutting out sugar and cutting out, so I don't know so much about the science behind which foods are inflammatory or pro-inflammatory and which ones are not. I mean,
Mhm.
there's certain supplements or things like turmeric and so on that are,
Right.
data around their inflammatory properties, but people, a lot of people really swear by them. They feel that it's been
Yeah.
beneficial.
Yeah.
Um, and I wonder about if you, I mean, I'm semi, I guess there's no, it's probably an oxymoron to say I'm semi-vegan because that means I'm not a vegan. But I do like to eat more plant-based foods and um but I wonder about protein intake, whether one has to, pay particular attention to how much protein you're consuming, especially if your muscles are being challenged by having FSHD, and they might be in need of having more protein to just kind of help maintain them
I
better.
mean, you definitely have the pea protein whey. You have that stuff that you could also take. But then even just having, making sure you have plant-based stuff that is full of protein. So everything from like your quinoas to your beans, your peas, and getting adequate amount of that, your dark leafy greens and stuff like that. You know, don't try to be vegan and be trendy. You have to make sure, like you said, you get your protein.
Yeah, no, yeah, you follow sound nutritional principles.
Yeah.
Yeah. So,
And I tell people, when from an eating standpoint, whether it's FSHD or colitis or whatever, right? From an eating standpoint, you want to try your best to figure out all the different things that can cause inflammation. Do like just a massive Google search, right? And speak to your nutritionist, speak to your doctors as to what foods tend to cause inflammation, and don't just take them out of your diet one at a time.
mhm.
Take a bunch of them out at a time and then reintroduce them back into your diet one at a time. So for instance, if let's say milk and I don't know, apples and peppers cause your symptoms, right? And if you say, okay, well, let me stop taking peppers, but you keep taking your apples and your milk, you're still going to get inflammation, and you're going to say, oh, well, what I took out isn't, that isn't the problem. So you put it back in, and then you take out something else one at a time. Like now you take out the I don't know, the peppers, and that you're still getting inflammation because you're still taking the other stuff. So if you were to take them all out at the same time, you reintroduce them one at a time, at least then you have a clearer picture as to, oh, this in particular is causing me issues. As opposed to it's being masked because you're still taking other things that could be causing issues, if that makes sense.
Yeah, no, that totally does make sense. Oh my gosh, I
Yeah,
just realized we are well after the hour. I'm really sorry, but
that's
um,
when the conversation's good.
yeah, it is a good conversation. But I want to be mindful of people's time as well. So I
Yes, yes,
guess we
yes,
could
yes.
formally um, let Eloise go because she has probably several exams or something or papers and things she's got to work on. And I need to go because I have other things I need to do, but I can definitely keep this open if people want to continue conversation. I will give Zach I'm going to
Post. Oh, I was just gonna mention also, if anybody wants to reach out with any additional questions, I'm gonna put in my email into the chat in case any questions pop up at any point.
Cool.
Really appreciate you taking the time in for the research effort too.
Okay, I just sent it. Yeah, well, thank you all so much for joining. And Zach, if you want to close it out.
But yeah, I was just saying, appreciate you taking the time and for your research efforts, and thanks everybody else for joining too.
Of course, thank you all for being part of the community.
Have a good night, everybody.